My name is Curtin Conrad. I'm a dermatologist in Lausanne, and together with my co-chair, Katia Boniface from Bordeaux, it's really a great pleasure to welcome you to this month ESTR Kitchen, which is a molecular cuisine. That means we're having a really well-known leader in dermatology, investigative dermatology, who will talk about discovery pathway during their career. And today, I think it's a perfect timing because yesterday we had the International Day of Women and Girls in Science. And today we really have a shining example for such a woman in science with Sabine Eming. So Sabine Eming is a professor of dermatology at the University of Cologne, where she also received her dermatology training. Then she was a research fellow at the Scripps Institute and the Harvard Medical School in Boston. And her research interests over the last years, I wouldn't say decades, but years, on how the skin senses damage and how it reacts into a regenerative response and in disease. And their findings have really led to discoveries that had influence in the treatment of such diseases and had also influence on how we see other diseases such as diabetes mellitus, but also skin aging and wound repair. And for all her work, she received several or numerous awards and prizes, including, for example, the Oscar Gons Award from the German Society. And last but not least, I think one of her big achievement was she is a past president of our society, of the ESDR, and she's actually the current sitting Secretary General of the ISID and will be so for the upcoming meeting in 2028 in Europe. So Sabine, it's a great pleasure that you have accepted our invitation to give this lecture here, or this kitchen webinar. And I'll just shut up and hand you over the microphone. Looking forward to your talk. Thanks a lot.
So, dear friends and colleagues, so thank you very much, Curtin, for the very kind and nice introduction. Also to you, Katia, for the invitation. It's a great pleasure, actually an honor to be here and to present, to have the opportunity to tell a story in this kitchen series. Yes, and as you just mentioned it, for a long time, my group and I have been interested to understand how the skin and tissues, how they repair and what goes wrong if they lead to a misregulated repair response. And I think it's a fascinating process. And that is actually the goal of my lecture, also to bring this over, to bring this across. And even to go maybe even a little bit further to say that a misregulated wound healing response can lead also, or can be the underlying disease mechanism for maybe your preferred disease. So if at the end of the talk, you go back and look at your preferred disease that you investigate and think, so maybe the underlying pathology could be, there could be something from a misregulated wound healing response there, I think I reached my goal here in this lecture. So just to start, what are our major challenges, or some of the major challenges in medicine? I mean, to those contribute scar, fibrosis, and also cancer. I mean, some of those we are able to treat already better than maybe 20 or 10 years ago, but certainly what is still a major problem is tissue fibrosis. And very often tissue fibrosis leads to loss of tissue function that then even precedes cancer or even carcinogenic situations. So, and as we know, I mean, these situations contribute significantly to morbidity, and they are also of major cause of socioeconomic burden. Meaning, if we better understand the underlying pathologies, or maybe a common pathology of this situation, then we could or would be able maybe to tackle and to have an impact on morbidity and socioeconomic burden, to reduce those. And it is not easy to find statistics and data on numbers on the prevalence of fibrotic diseases. And I just wanted to briefly point out to you this report that I found on clinical epidemiology. And this is basically a study which was performed in the UK. It was based on electronic healthcare record data. And here the authors analyzed prevalence of situations where basically in the healthcare documentation, there was the word fibrotic or cirrhosis. And here they took roughly 1 million UK adults. And what they found is that there is a prevalence, and the study was performed in 2015, of around 20% fibrotic conditions in the general population. And 6% of those had even several situations of a fibrotic disease. So the point is here basically that the number is high, and it is certainly important that we understand underlying mechanisms and certainly also better how to treat those. And we as dermatologists, of course, we know very well what scarring and a misregulated wound healing response means. And just to bring this together, I just show you this slide again, which basically shows us the clinical spectrum of skin wound healing, which during lifetime can be very different. So we start off even prenatally. This is the best, this is a perfect repair response, because there we heal scar-free. This is true regeneration. But then after birth, we lose this capability. And as you can just see here, of course, any injury that goes beneath the epidermis will leave a minor or major scar. And then we have pathologies like hypertrophic scarring or fibrotic diseases, ulcerative impaired healing. And we know that chronic non-healing wounds always have the danger and the possibility even to develop a cancer. So basically what we see here is that this situation here at the beginning, I mean, this is the best how it can get our healing response after birth. And even so, that is already fantastic that we can quickly heal and seal wounds. But at the end, it is a story of a misregulated, more or less misregulated wound healing response. That certainly can lead to disease. And this is the situation which we see common. But I think also here are examples, and I just wanted to present to you the dystrophic EB or UV damage here in an elderly patient, where we know very well, and these are very common examples in our daily clinic, where skin fragility and small injuries initiate a wound healing response, which can be efficient maybe at the beginning, but it will be repetitive, right? And we have a circle of skin fragility, injury, wound healing, and which then also at some point, if you repeat this several times, it will lead to chronic inflammation. And it will lead to fibrosis, tissue stiffness, which then even will impact genome instability and might lead to cancer. So at least this, I think, is the current understanding what these two conditions, which I show here, or even the one before, might have in common. And the question is, can we understand what goes wrong, or when the physiological healing response deviates from physiological healing into pathology? And can we, of course, prevent it and interfere? That is the big question. And just briefly to remind you to the physiology and also the pathology of healing, I mean, this process is now quite good understood. So we have a highly dynamic response of injury or after the injury of an inflammatory response, as we see here. Innate cells, immune cells are involved, the vasculature, cell death processes. We have a highly inflammatory situation. We have this phase of tissue proliferation, endothelial cells, and also fibroblasts, tissue resident cells are activated. And then what is very important is, of course, as soon as the epithelium is closed, we have a dramatic alteration in cellular processes, and there has a resolution process, right? I mean, the inflammatory response will decline or will significantly change. We have a clearing process, macrophages perform the phagocytosis, change the activation state, interact differently with tissue resident cells, and we have this resolution phase, which is important and which then, of course, is all embedded also into an extracellular matrix response. But here we have more the cellular view on it. So what goes wrong? What we know is the situation. So either, I mean, basically the simplistic understanding is that the healing response will not follow its dynamic. So it either gets stuck in inflammatory cycles, which are detrimental to the tissue, which cause ulceration, tissue damage. Or we can have even the situation where we have a major problem in the later stages, where we have an increasing fibrotic response, fibrotic circles, which lead to tissue fibrosis, and also a chronic inflammatory situation. So as just said, at the end, we have to understand which are the underlying signals and molecules. And hopefully, with a better understanding, we will then also be able to understand where is the deviation, where the physiological response either goes to these inflammatory cycles or to profibrosis. So the question basically is, what is upstream? Upstream of this, let's call it a misregulated repair response, where at the end I will have scarring, fibrosis, or cancer. And it is our hypothesis that this might be the immune response. So our question is, so is it an inappropriate immune control, which is really the shared catalyst, which is upstream for this dysregulated repair response and then downstream pathologies? And how do we come to this hypothesis, or what is the basis? And here just focus and concentrate on different outcomes of the repair or regenerative response. So here we see on the left side that axolotl zebrafish, they can heal very well. They are the paramount examples of tissue regeneration. Right? I think we all know there is examples of a damaged arm of the axolotl, which entirely can regenerate in several weeks. And what is also remarkable in these creatures is that they are resistant to disease. For example, in axolotl, you cannot induce cancer or a neoplasm. And on the other side, of course, we have the mammalian system, which is prone to scar formation or scar-based healing. And this goes along, of course, with susceptibility to disease. And what has become clear over the past years is that immune signals are the upstream signals and mediators that decide whether a tissue can perfectly regenerate or forms a scar. And that this is also associated, as just pointed out here, with disease resistance or whether it comes to disease. So therefore, we think it is critical and important to understand which immune signals decide whether a tissue will regenerate or whether it will form a scar and then go even and can form a disease situation. Of course, the immune system is complex and many signals are important. For now, here I would like to bring the focus on myeloid cells, particularly on macrophages. So others and also our group, for a long time now, we investigated the role of macrophages, the different phases, the many different phases of macrophages in injured tissue and also in healing tissues. And let me just briefly summarize what we have found here. So first of all, macrophages are key drivers of wound healing. So without macrophages, there will be no healing response, right? And very important, there will be no resolution of the inflammatory or damage-induced inflammatory response. So, and in skin here, of course, we know that after the injury, we have this highly pro-inflammatory situation, which is critical also for host defense. We know that macrophages are critical mediators also of trophic functions. For example, the angiogenic response and sprouting vessels requires and depends on macrophages. And on the other side, we also know that resolution and matrix formation depends also on macrophages. And as you can see here, when we see these early pro-inflammatory mediators, interleukin-1 and interleukin-6, I mean, here we see that also late stage, when the epithelium is already closed, there is still an immune signal in interleukin that drives this resolution process. So it's a different quality of the immune response. And here we know that type 2 cytokines are important. And we have shown this, we have published this, so I will be very quick. So basically what we have shown several years ago is that type 2 activated macrophages, that they are critical to form the scar and that they interact with fibroblasts, release certain mediators. Here is the resistin-like molecule that then initiates or it leads to the expression of enzymes that are critical for collagen cross-linking. And here it is the lysyl hydroxylase 2. And just very quickly, what have we seen? In mice that were damaged, and here we have mice that lack the interleukin-4 receptor on macrophages. Basically, here at day 7, here's the control. Here's the knockout. We saw that these tissues are highly hemorrhagic. We saw that the matrix collagen fibers were significantly different than in a normal healing wound. And we saw also that biochemically cross-linking here in the red dots was altered and was not as efficient as in the control. So in our assumption here was that basically the matrix is not stable, is mechanistically not stable, not robust in these mice, and therefore also vascular stability is impaired, which could explain this hemorrhagic phenotype. So basically it shows us we need type 2 activation in late-stage macrophages for efficient wound healing, at least scar-forming healing, which is the physiological situation in skin. So, and that motivated us to better understand, so which are the mediators that drive this reprogramming of macrophages from this very inflammatory stage to this rather resolution type 2 activated repair stage. And to better understand this, we performed a single-cell RNA-seq analysis of wound tissue in early stage and late stage wound macrophage in mice. And this work was primarily done by Sebastian Willenborg. And it has been also published, so I will be quick. Basically, what we see here is that we do not have one early and one late stage macrophage. I mean, macrophage activation is very heterogeneous. So in this study, we found basically that macrophage activation cluster into 7 clusters. And here we see that, for example, cluster 2 and 3, the green ones, are enlarged in early stage and they reduce in late stage. And in addition, what we see is, for example, that cluster 0 or 6 increases at late stage or even appears newly. So the point here is that we have a large heterogeneity and that we have to understand this heterogeneity. And we see, for example, here that basically we could confirm what we saw already also in earlier studies, that the early macrophages are highly pro-inflammatory type 1, and the late stage have a clear type 2 signature, a resolution signature. And then the question was, so can we further assign molecular signatures to cell function? And what we found here is, for example, that the early ones, these pro-inflammatory, are highly glycolytic, and the resolution macrophages have an entirely different metabolism. They are in the OXPHOS mode. So the question that we then had was, is this just a bystander effect? Or is maybe the metabolic state also in somehow relevant to regulate the activation state and execute effector functions of wound macrophages? So here you just see again genes that were highly upregulated in early ones, which are all relevant for glycolytic metabolism. In the late stage macrophages, you see many genes upregulated, relevant for lipolysis, fatty acid oxidation, which is all relevant in OXPHOS metabolism. And here, even when we show these activated macrophages in vitro, and Astrid shows here also, she showed the different morphology of mitochondria, which drive our metabolic state. They are fragmented in the glycolytic macrophages, and here we see in the OXPHOS macrophages in OXPHOS mode, they fuse, they form tubules, they have an entirely different morphology. So our question here is, so is this different metabolic state, does it have any impact? Is it relevant for fibroblast activation? And at the end, even scar formation? And always the question is, of course, can we interfere? So, and the question which we then had was, so is it possible that metabolites and specific mitochondrial metabolites might control immune cell function? And this is, of course, not our own idea. This is now an idea which is investigated for 8 or even 10 years. That it became evident, and innate immune cells were the first, where it became clear that it is metabolites that can drive cell function. And that is actually what we asked. And always when we enter a new field, of course, we try to review the literature, what is known. And here I had the opportunity to review basically which Krebs cycle metabolites have been already known to have an impact in the healing response. And then we found, for example, we know that these early macrophages are in this Warburg metabolism. And of course, one of the oldest metabolite that is studied in wound healing is lactate, right? Which is highly produced here also. It's one of the strongest inducer of the angiogenic response. And just recently, it has been found that also succinate is very important, which is highly up in this pro-inflammatory situation. It drives and controls HIF-1 stability. On the other side, when cells are in OXPHOS metabolism, we have an upregulation, for example, of the alpha-ketoglutarate. And alpha-ketoglutarate is a cofactor for many enzymes and mediators that I mentioned already to you and which are required for resolution. For example, alpha-ketoglutarate is required as a cofactor for the lysyl hydroxylase 2, which I just showed you before, important for cross-linking. And the gene is PLOT2. Or, in addition, alpha-KG is also relevant cofactor for histone demethylases, which are required for the reprogram of early into late stage macrophages. And just to see this and to see how it correlates maybe with the genes that we also found in the transcriptomes. Here we see, for example, gene upregulated for an important histone demethylase, also in late-stage wound macrophages. Or here the isocitrate dehydrogenase, which is basically the enzyme responsible for the generation of alpha-KG. So this is correlative, and the question is, can we show that it is functionally connected? Here I just show you, we just go back to some wound data of wound macrophages, and certainly to see whether all this transcript data also results in function. We performed metabolic analysis. Here we have a seahorse analysis. We see that early wound macrophages are indeed highly glycolytic, and the late ones use a lot of oxygen. They are less glycolytic. So consistent with the transcripts I just showed you before. And this also goes in parallel with an increase of the mitochondrial mass. Then the question is, does this really depend on type 2 signaling on cytokines? So here we performed the same analysis in macrophages deficient on interleukin-4 receptor alpha. And here we see that late-stage wound macrophages have significantly less oxygen consumption. So supporting the idea that type 2 signaling is important to drive late-stage macrophage fibroblast crosstalk, and eventually is also relevant in scar formation. So then the question is, can we confirm this, and do we see this also on the metabolites? And here we see, basically, we performed a semi-targeted metabolic analysis in wild-type bone marrow macrophages stimulated either with LPS interferon or with type 2 cytokines. We clearly see what I showed you before. We have an upregulation in succinate in the early ones, alpha-ketoglutarate in the type 2 activated macrophages. And important is that we have here the ratio of alpha-KG and succinate, which we clearly see up in the late-stage macrophages. And I see that I'm already close to the end. Is it true? Yes. So I will finish in 2 minutes. I will count. So then I have to go over some slides quickly. But basically what we show here is that we have shown you on transcript function. That is also what we see really in metabolites. And the question, of course, was then, so can we interfere here functionally? And we generated mice where in macrophages, basically we delete COX-10, which is a factor that is required to assembly complex 4 in the electron transport chain. So these macrophages are basically deficient. They cannot go into OX-phos metabolism. And I will go now quickly over this. So here we have shown that this is possible. For time reason, I have to be quickly here. Important is, for example, what we showed here, that these cells, when we analyze metabolites, for example, stimulate them with type 2 cytokines, the controls go up, as I just showed you before, but the COX-10 deficient macrophages cannot do this. Showing very efficiently, these cells cannot produce sufficiently alpha-ketoglutarate because they cannot go into OX-phos metabolism. Does it have an impact on the healing response? I show you this slide. We are here at day 7 after injury. We stain for myofibroblasts, and we see that myofibroblasts are abundantly present in the control mice, but we have a clear deficiency here in the COX-10 knockout mice. So meaning that you need OX-phos capable macrophages to communicate with fibroblast for the induction of myofibroblast formation. Basically, that is the hypothesis. And just to move on, basically what we have shown and what is important to understand now, what is the problem in the COX-10 deficient macrophages. And here we see, for example, they are highly pro-inflammatory. They cannot reprogram. Basically here the late stage mediators, type 2 mediators, RELM and CD206, they are missing. So these macrophages basically cannot reprogram. You require a functional ECT to reprogram from the inflammatory to the OX-phos resolution stage. And let me just quickly go through this. So basically the idea here was or the hypothesis basically is it possible that a specific subset of macrophages is missing that has been described earlier from Valerie Horsley that is critical to induce a specific subset of fibroblasts, the adipocyte precursor fibroblasts, which are required to form a fibrotic tissue. So, and maybe I just go now to the summary slide where we have our hypothesis. Basically, what we could show is that exactly in the COX-10 knockout mice, we have this population missing, which regulates or which reduces, releases mediators such as the PDGFC or IGF-1, which Valerie has shown to be critical to induce myofibroblast differentiation. And our idea here is, or the hypothesis would be that you need type 2 activation, you need a functional OXPHOS metabolism, you need a high alpha-KG and succinate ratio to have this happen, because that is basically what we saw defective in our mouse model. So the idea would be, so this is in physiological healing. And then the idea is, of course, that when you have too much, when you have an uncontrolled and increased type 2 immunity, whether that could be a target also in fibrotic diseases. And here I just showed you several situations. And we know that type 2 immunity, I showed you activities for macrophage activation, metabolism, but there are many more. So basically the idea is whether we can interfere and improve the outcome. And we just finished now a study where we treated patients with morphia, plaque morphia, with dupilumab, blocking type 2 activation. And we now have the outcome and now know that patients indeed benefit significantly from dupilumab in this situation. Of course, it needs a larger population to confirm this, because this was just a smaller phase 2 study. Finally, I would like to thank colleagues, everybody who was involved in this, particularly Sebastian. He gets great support by the group. But this work was certainly only possible with a fantastic collaboration here on the campus, basically with our colleagues, which are specialists in mitochondria biology. And on the other side, also our colleagues, of course, which are expert in type 2 immunity. And to have both in our group, I mean, that allows us to come to these conclusions. So thank you very much for your attention, and I'm happy to answer questions.
Thank you, Sabine, for this really interesting and exciting talk about wound repair. So please feel free to ask your question in the Q&A. So I see we already have one question that I'm going to read. So the different macrophage clusters you detected in the skin over the course of healing, are they there all the time, or do they change their phenotype due to plasticity in response to cytokines and cell-cell interaction?
Well, that's a good question. And so now we think macrophages in this model, which goes over 2 weeks, that a large portion of the macrophages, which we see at the late stage, stays, which are apart from the initial ones. And they change due to external factors, as just shown. I mean, the type 2 signals, they have to come from the tissue. But in addition, of course, we have a mixture also of the ontogeny of the macrophages, right? The majority is recruited. These are the blood-derived monocytes that switch the activation states. But in addition, of course, we have also tissue-resident macrophages. So we do not know exactly the difference between these different types of macrophages at late stage. But we are sure, and actually we have shown this also by tracing studies in the cell metabolism paper from 21, that cells will, significant portion of the cells will stay over these 2 weeks.
Thank you. So I have a question. So regarding your COX-10 results, so in human, so diseases or like people who have like repair alteration. So maybe you showed it and I missed it, but is there a regulation of COX-10 expression? And do you think it could be a target actually to modulate this expression?
Well, our studies here is, I mean, we are at the stage of proof of principle.
Yeah.
So I'm not aware, there is not a disease where you have specific COX-10 mutation or deficiency.
Okay.
Of course, there are patients which have alterations in their mitochondria function. But usually these patients have other more severe diseases. They are compromised, right? In muscle activity, in neurological diseases. So, and there could be also compensatory mechanisms, of course. So here at the stage, we are on proof of example, proof of principle, whether by changing metabolism, we can impact cell function and also tissue function, I mean, the outcome of tissue function. And I mean, we have not yet looked into, for example, fibrotic tissue to see whether there is also an altered metabolic signature. Mm-hmm. But now with the data from the Dupimorph study, we do have transcriptome data, and I think that will be very interesting for the next weeks to come to investigate that.
Okay.
I think it's just maybe a question, a bit follow-up to the first one, timing being important. So, you know, Th2, 22 cytokines might be important for repolarization. Then IL-4 seems to be more important for the fibrotic part. Could it be that there's a timing which is important and depends when which signal pops up and how it changes?
Yes.
Because in the same regard, there have been some cases described that dupilumab can induce some Morphea-like injuries, which would be very paradoxical to your story.
Yes. I mean, there have been one or two case reports, right, which reported on fibrotic diseases occurring. So, I mean, these are case reports. So here we at least have 45 patients in our trial, so which is already a little bit more. And there is really a significant correlative descriptive data that shows that with fibrotic diseases, you have an increase of a type 2 signature. Just coming back to your first question, I mean, timing is important. I mean, very sure. And I think what will be also important, probably which cells deliver the type 2 signature or the type 2 signals within the tissue, right? It will be mast cells, it will be eosinophils, it will be ILCs, which all have a different dynamic in the healing response. So timing and also the niche where macrophages or other cells might be activated and are in contact with type 2 signaling cytokines and producing cells are presented, that will be, I think it will be very important.
Well, thank you. We are running late. So thank you very much, Sabine, for your talk. It was really interesting. Looking forward to the publication soon, maybe with all these results. And so the next.
Thank you very much.
No, thank you. Sorry. Yeah. So the next Yes Your Kitchen webinar in March will be a sweet and sour and vitiligo. So with Mauro Picardo and Julien Sénéchal. And also, so today's webinar, you can tell your colleagues that will be, as the other ones, available on the YouTube channel. And so lastly, maybe Leah, if you can put, yes, the next slide. Also, please, the next slide. So now the applications for the Futures Leader Academy are open. So since the beginning of the month until March 17th. So please encourage the youngest and promising researchers in skin biology to apply to this FLA. And it will happen in France, in Nice. So hopefully it should be a very nice weather. So with a lot of really famous and well-renowned chairs and mentors. So thank you all for being here today. Thanks again, Sabine.